Hello students, in this video you will be studying about photosynthesis in higher plants. Green plants carry out photosynthesis, which is a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. All living forms on the earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. So photosynthesis is very important due to two reasons. It is the primary source of all food on earth, and it is also responsible for the release of oxygen into the atmosphere by green plants. Cornelius van Niel, based on his studies on purple and green bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable compound reduces carbon dioxide to carbohydrates. Later, Robert Hill demonstrated that the oxygen released into the atmosphere comes from water. The process of photosynthesis takes place only in the green organs of the plants. Amongst these, the leaves are the main organs. Even in the leaf, this process occurs in the chlorenchyma of the mesophyll. And the mesophyll cells possess organelles called chloroplasts. And the chloroplasts are arranged in the peripheral region of the cell, which facilitates the diffusion of gases. Within the chloroplast, there is the membranous system consisting of the grana and the fluid part, which is the stroma. Each granum is made up of flat lamellar structures, which are the thylakoids, and the thylakoids are arranged like a stack. And these thylakoids contain the chlorophyll pigments. The membrane system is responsible for trapping the light energy and synthesis of ATP and NADPH2. And this is called the light reaction. And in the stroma, enzymatic reactions incorporating carbon dioxide into the plant, leading to the synthesis of sugar. Since it is not a light driven process, it is known as the dark reaction. Chromatographic separation of leaf pigments shows that the color that we see in leaves is not due to a single pigment, but due to basically four pigments: chlorophyll A, B, xanthophyll, and carotenoids. Chlorophyll A and B are green pigments, xanthophylls are yellow, and carotenoids are orange pigments. And these pigments are concerned with the absorption of light energy. They absorb light at a specific wavelength and obtain energy from them. This energy is first converted into the energy of electrons and later into the chemical energy. Chlorophyll A molecules are the main reactants in this process. The other pigments direct and focus the energy absorbed by them towards the chlorophyll A molecule, and hence these other molecules are called as the accessory pigments, and the chlorophyll A molecules act as the reaction centers. Now we will study about the light reactions. Living organisms have the ability of extracting energy from oxidizable substances and store this energy in the form of bond energy. And special substances like ATP carry this energy in their chemical bonds. So the process through which the ATP is synthesized by cells is called as phosphorylation. And photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first the pigment system 2 and then the pigment system 1, a process called non-cyclic photophosphorylation occurs, and the two photosystems are connected through an electron transport chain, and both ATP and NADP H and H plus are synthesized by this kind of electron flow. Now in case of the cyclic photophosphorylation, only the pigment system 1 participates. The pigment system, which is made up of many chlorophyll molecules except one molecule of chlorophyll A, forming the light harvesting system, which is called as the antennae, and the single chlorophyll A molecule forms the reaction center. Now we will study about the light reactions which are taking place in the thylakoid membranes of the chloroplast. And the light reactions use two photosystems. The photosystems are the photosynthetic pigments, which are organized into two photochemical light harvesting complexes, which are the two photosystems PS1 and PS2. In the first one, that is PS1, the chlorophyll A molecules at the reaction center are stimulated by the wavelength of 700 nanometers, and so it is called the P700. And in PS2, the chlorophyll A molecule at the reaction center are stimulated by the wavelength of 680 nanometers, so it is called the PS2 or P680. And each pigment system is made up of around 250 to 400 molecules of chlorophyll molecules, which form the light harvesting system. In photosynthesis, two sets of reactions are taking place. The first one is the light reactions, which are light dependent and energy capturing, and the second group of reactions is the Calvin cycle, which is a light independent reactions. Pigment system 1 is stimulated by absorbing the light energy of wavelength 700 nanometers. So, 4 energy rich electrons are freed from the reaction center of PS1, and these electrons are picked up by the electron acceptor, which passes them to an electron transport system consisting of the cytochromes. And during this course of electron transport, energy is released from the electrons, which is utilized for the synthesis of two molecules of ATP. As the electrons return to their original source, then the electron transport system is called cyclic electron transport. In the second type, that is the non-cyclic photophosphorylation, the pigment system 1 and pigment system 2 both take part, in which 4 photons of light of 680 nanometers of wavelength stimulates pigment system 2, and 2 electrons are released from the reaction center. And these 4 electrons are accepted by various electron acceptors and finally entering into the pigment system 1 instead of returning to the pigment system 2. So the chlorophyll A molecules at the reaction center of pigment system 1 receives 4 electrons from the pigment system 2. And during this transport of electrons, energy is released which is stored in the phosphorylation of ADP to ATP. And simultaneously 4 electrons which are released by the reaction center of pigment system 1 after receiving light of wavelength 700 nanometers neither go to pigment system 1 nor to pigment system 2 but are accepted by the electron acceptor and then utilized for the reduction of NADP. As the electrons released in various ways do not return to their original donors. Such an electron transport is called non-cyclic electron transport. Here you can see from the photosystem 1 which accepts the light energy and releasing the electrons which are accepted by the primary electron acceptor, and then it comes back to the same photosystem 1. So this is the cyclic photophosphorylation. And in non-cyclic photophosphorylation, photosystem 2 regains electrons by splitting water, leaving oxygen gas as a byproduct. So from photosystem 2, the electrons are coming and which are accepted by the electron acceptor, and it passes through the electron transport system and finally it reaches to the pigment system 1. So it is called as the non-cyclic photophosphorylation. The splitting of water molecule using energy of light is known as photolysis of water. During this process, 4 molecules of water split simultaneously, and for H plus and for OH minus ions are released. These are accepted by the hydrogen acceptor NADP and getting converted into NADPH2. The OH minus ions lose their electrons and release 2 molecules of water and 1 molecule of oxygen. And manganese ions, chloride ions, calcium ions play important roles in this process. So the light energy required for photolysis of water is absorbed by the pigment system 2. Now let us try to understand how ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain this mechanism. ATP synthesis is linked to the development of the proton gradient across the membrane. The entire biosynthetic phase or the Calvin cycle can be described under 3 steps. The first step is the carboxylation. Carboxylation is the fixation of carbon dioxide into a stable organic intermediate. First of all, the carbon dioxide which is obtained from the atmosphere combines with 5-carbon pentose sugar, which is the ribulose biphosphate, which is obtained from the stroma, and a 6-carbon unstable complex is formed, which splits into 2 molecules of 3-carbon compound, which is called as the PGA or phosphoglyceric acid. This reaction is catalyzed by the enzyme RUBP carboxylase, ribulose biphosphate carboxylase. So this first phase is called the carboxylation phase. Then the second phase, which is the reduction phase. A series of reactions are involved in the formation of glucose in the second step. The steps involved utilization of ATP molecules, 2 molecules of ATP for phosphorylation and 2 molecules of NADPH for reduction per carbon dioxide molecules fixed. For the formation of 1 molecule of glucose, fixation of 6 molecules of carbon dioxide, and 6 turns of the cycle are required. And the third and the final step is the regeneration stage. Regeneration of ribulose biphosphate, which is essential if the cycle is to be continued without any break. So 1 ATP is also required for phosphorylation to form the ribulose biphosphate, for the regeneration of ribulose biphosphate. So in this way, for every carbon dioxide molecule entering the Calvin cycle, 3 molecules of ATP and 2 molecules of NADPH are required. And for the formation of 1 molecule of glucose, 6 turns of the cycle are required. During light reaction, ATP, NADPH and oxygen are produced. Oxygen diffuses out of the chloroplast. ATP and NADPH are used in the process of dark reaction or the biosynthetic phase, which is not depend on light, but it depend on the products of light reaction. Now in the biosynthetic phase, water combines with the carbon dioxide to produce sugar. The scientists took a long time and conducted many experiments to know the primary acceptor of carbon dioxide. Melvin Calvin and his co-workers worked out the whole pathway and showed that the pathway operated in a cyclic manner, and the entire Calvin cycle or the dark reaction or the biosynthetic phase, it is called. The ATP produced will be used immediately along with NADPH in the biosynthetic phase, which is taking place in the stroma of the chloroplast. The proton accumulation takes place in the lumen of the thylakoid. As the splitting of water molecule takes place inside the thylakoid, the protons accumulate within the lumen of the thylakoid. And on the other side, the reduction of NADP takes place in the stroma, which is outside the thylakoid membrane. And for this reduction, protons are required, and the protons are present in the stroma, which are used up. So within the chloroplast, protons in the stroma decreases in number, and in the thylakoid lumen there is accumulation of protons, and this creates a proton gradient across the thylakoid membrane. And it is the breakdown of this proton gradient which leads to the release of energy. And this gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channels of the F0 of the ATPase. And the ATPase enzyme is consisting of two parts, the F0 part which is embedded in the membrane and which forms the actual transmembrane channel, and F1 which protrudes on the outer surface of the thylakoid membrane on that side that faces the stroma. And the breakdown of the gradient provides enough energy to cause a conformational change in the F1 particle of the ATPase enzyme.